would want to thank you for taking the time today to join us to learn about the latest in MS research. This event also gives us a chance to connect with members of the MS community and learn more about a disease that affects more than 90,000 people in this country. The two online conference, which brings together people affected by MS, healthcare professionals, and researchers from across the country, allow us or also helps us to prepare for MS Awareness Month, which is fast approaching. I'm very pleased to have the opportunity to hear Dr. Alexandre Prat speak to us this afternoon about his mark on biomarkers. This area of research holds great promise for the diagnosis and treatment of MS and is a source of hope, especially for people living with progressive MS. Before I introduce our speaker, I would like to provide you with some useful information. You can submit questions at any time during the conference using the box provided. You can join the conversation also online and share your favorite moments from today's presentation by posting a message on Twitter with a keyword #ConnectionSP. So you can have your say also by taking our daily survey on our Twitter account at Society MS Canada. This presentation is being recorded and will be posted on connectionsm.ca. I now have the honor to introduce our speaker, Dr. Alexandre Prat. Dr. Prat is a neurologist at the CHUM in Montreal and a full professor of neuroscience at the University of Montreal. After completing a PhD under the supervision of Dr. Jack P. Antel, he completed his residency in neurology at McGill University, specifically the Montreal Neurological Institute until 2003. In 2000, he received the prestigious S. Weir Mitchell Award from the American Academy of Neurology. So welcome, Dr. Prat.
Good morning or good day. Good morning or good day. My name is Alexandre Prat. I am a neurologist at the CHUM in Montreal and researcher at the University of Montreal. The MS Society of Canada asked me to speak to you in French today. I could have done this in English as well. And I will speak to you now about a few pretty recent developments in fundamental research and clinical research. The title of my presentation will be A New Technology to Identify Biomarkers of the Trajectory of the Multiple Sclerosis Disease. I am a researcher at the University of Montreal, as I said, and I am also the holder of the Canada Research Chair in Multiple Sclerosis. First of all, a few general facts about multiple sclerosis and the disease, although you probably know as much as I do on this. When I give courses in university or when I make public presentations, I always start by asking, who knows someone who has multiple sclerosis? And systematically, the vast majority, if not the totality of participants, raise their hands. Everybody knows someone that's been affected by multiple sclerosis. Multiple sclerosis is a very heterogeneous disease. It's probably the most heterogeneous disease in medicine. Heterogeneous in its terms as its form of presentation, heterogeneous in terms of the age of presentation, in terms of its progression, in terms of response to treatment. It's a disease that is totally heterogeneous. There are approximately 650,000 North Americans affected by MS and probably 100,000 Canadians, including 20,000 who don't know. The average age at diagnosis is between 25 and 29, as we know, and it's a disease that affects women more often than men, 3 times more often women than men. But the disease is sometimes a lot more severe when it shows up in a man, although not always. We say more often, more severe in men. The disease shows up initially in terms of relapses and remittances in 90% of patients. And after 10 to 15 years, if there's been no treatment, half of them will develop a progression of the disability. What we know through new studies, but also from our clinical experience, is that the entry of treatments in the therapeutic arsenal in multiple sclerosis delays. So when you start taking medication, it delays considerably progression. Sometimes the attacks are severe and very disabling, but most of them are resolved easily and leave no trace. What patients and neurologists fear the most is progression. So as I said, here is a little schematic diagram reflecting the evolution of disease over time. On the x-axis here, you see time that goes by, and here on the y-axis, you see the severity of the disease. At the beginning, there's a long phase which is asymptomatic. We believe that the asymptomatic phase, during which patients have no symptoms, could last 10, 15, or even 20 years. And then the patient starts to feel attacks, which are shown here by this bar, then a plateau, and a few weeks or months later, a spontaneous remission, going back to normal. And that can last 10 to 15 years. Then there is less recovery between the attacks, and finally there's an increase of disability over time. I'll give you a brief example. This is a young woman, a nurse in our hospital, who comes to a consultation one day with symptoms that are extremely typical of MS. Look here. I ask her to look to the right. See that her right eye can move to the right, but not the left eye. I ask her to look to the left. Her left eye moves to the left, but not the right eye. When she looks upwards, the look is totally disconjugated between the two eyes. We put her on a medication, on a treatment, and a few months later, a few weeks later, she recovered completely. She's totally back to normal. Look at this. Both eyes move perfectly. Both eyes move perfectly on the other direction and upwards. She's had 2 kids since now, maybe 3, and she's never had any other relapse since she's been treated with a drug, which I will not name, of course. Now, how does this disease show up pathologically? It's through the formation of plaques, sclerosis patches. This is the brain of a patient with multiple sclerosis, and that patient gave his brain for research. So you see with the black arrows and the white arrow, you see patches or plaques located in what we call the white matter of the brain. There are also plaques located at the junction between white matter and gray matter here at the top. Those are plaques which we rediscovered more recently, just about 10 years ago, thanks to all the new studies and the new breakthroughs in magnetic resonance imagery, MRI. We know that in a normal brain, the brain contains only very few white cells, just a few white cells patrolling the brain for viral infections or to get rid of dead cells, but very, very few white cells. What you see here on this microscopy image is myelin, which is colored in blue, and the cells which are colored in pink. What happens with multiple sclerosis is that the white cells of the blood penetrate the brain and accumulate around the vessels, invading the brain, attacking and destroying the oligodendrocytes and neurons. You see on this microscopy image a lot of immunity cells located in the perivascular space, but that also migrated within the brain to destroy what used to be the myelin, colored in blue, and the oligodendrocytes. This myelin on the oligodendrocytes, as you can see on this little drawing, this myelin is totally necessary for the axons. The neurons and their axons can transmit the electrical influx from the cell of the neuron to the muscles. And if we no longer have myelin, or if it's been damaged, the flux can continue to flow, but much more slowly. The myelin is also necessary to keep the integrity of the neurons. If the neuron is demyelinated, if it lost its myelin, it will very often die. And that's what the white cells do when they enter the brain. They will attack myelin and, indirectly, the neurons. So these white cells in the brain of MS patients are made up of T cells, CD4 and CD8 T cells, macrophages, and more recently, as we discovered recently, B cells. And currently all treatments available to control the disease are treatments that act on T cells, B cells, or macrophages. I was telling you that multiple sclerosis is a very heterogeneous disease because of the diversity of its symptoms. Symptoms can be visual, loss of vision, such as an optic neuritis, or double vision, such as is the internuclear ophthalmoplegia or astaxis, which I showed you earlier on this young girl's video. There could be speech problems, there could be some sensory problems, numbness, lack of feeling, desensitization, pain. There could be motor problems with the muscles, although it's not the muscles that are affected, but the neurons that lead to the muscles, with paralysis, trembling, uncoordinated movements, and finally, urinary problems with urinary urgencies or difficulties to evacuate, or incontinence. There are very numerous clinical problems in multiple sclerosis. The most important problem for us is that it's terribly difficult to predict the trajectory for a given patient. Will the patient respond to treatment, for one? And if he doesn't respond, which of the patients will eventually progress and increase their disability? The other problem is that the progressive forms of the disease respond very poorly to the various treatments compared to the relapse, remittance, cyclical forms. There are very few specific treatments that are effective for the secondary progressive forms, and the current treatments have often a broad effect which is not specific for the molecular environment and can lead to important side effects. That's the reason why, in Canada, all the large multiple sclerosis groups decided to unite to start a study that is called CanPro-Co for Canadian Progression Cohort, or Canadian Cohort for Progression in MS. CanPro-Co is a synthesis of knowledge from the researchers at UBC, the University of Calgary, the University of Alberta, the University of Toronto, and the University of Montreal, to try to bring some answers to the issues that I spoke about. CanPro-Co is a large pan-Canadian study in which we recruited thousands of patients, including 500 patients at the beginning of MS disease. They will be followed during 5 to 7 years, all this with the purpose of determining with nuclear MRI, with immunology and neuroimmunology, and with epidemiology, what are the determinants of the progression in MS. We have co-investigators and collaborators everywhere in Canada, including Saskatoon, Winnipeg, Quebec City, McGill, the Memorial University in Newfoundland, and we will soon have completed recruitment of all the patients. So it's a study that is currently ongoing. The objectives of CanPro-Co are to, 1, discover new biological markers to predict the progression in MS thanks to a new technology, which I will explain, which is called the single-cell RNA sequencing. I'm sorry, we haven't yet been able to find a French translation for this new technology. And the second objective is to provide the research community, the MS research community in Canada, very high quality blood samples associated with MRI data and clinical data for use in the future. So in order to understand the single-cell RNA sequencing and the flow cytometry, which is the two new techniques we have, you have to understand what is a cell. You see in this drawing, you see a cell which is made up of a membrane that you can see here around the cell, and a nucleus. And in this nucleus, you have this famous DNA. When there is molecular production of molecules, the DNA splits apart in two parts, and it slides onto the DNA to transcribe RNAs, which are replicates of DNA. These RNAs will get out of the nucleus to get into the cytoplasm, where they will be translated once again into proteins. These proteins have many possibilities or many roles. They can be either secreted from the cell, so get out of the cell to act on other cells around. They can settle on the membrane of the cell to act as mediators, or they can enter into the composition of the cell and form the cytoplasm, the nucleus, and other enzymes. High-speed sequencing, which started in the years 2000, made it possible in 2003 to get a mapping of the whole human genome, which is now a reference framework for genes and proteins. And in 2006, this high-speed sequencing allowed us to start the sequencing of RNAs, and with a combination of microfluidic technology, allowed us to develop what we call single-cell RNA-seq. All the tissues in the organism, and by that we mean the organs. There's the brain tissue, the heart tissue, the lung tissue, the skin, the gastric tissue, and so on and so forth. All tissues are made up of millions and millions of cells, which are all attached to one another. If we can separate those cells, we can isolate the RNAs of those cells and determine exactly all the RNAs of all the cells that we can isolate. So with a single-cell RNA-seq, that's what we do. We separate the cells from one another, one by one, and we get all the RNAs of the cell in order to sequence them. So we know exactly what this cell will eventually secrete or produce as a protein. However, in flow cytometry, which is another technique that I want to talk about, what we do is that we identify the protein at the surface or inside the cell. So the single-cell RNA-seq is there to identify the RNAs, and the flow cytometry is there to identify proteins. The advantage of the single-cell RNA-seq over flow cytometry is that the single-cell RNA-seq identifies all the RNAs, whereas flow cytometry can identify, at this moment, only 21 proteins per cell. So in single-cell RNA-seq, you can identify up to 10,000 genes expressed by the cell, whereas in flow cytometry, we can identify only 21 proteins per cell. And by combining these two techniques with artificial intelligence techniques, you might know that Montreal is very strong in AI. It's a hub for artificial intelligence in the world. We have great AI experts in Montreal. We take all those information pieces. We have 40,000 per patient, for which we can identify 10,000 genes per cell. That gives you an astronomical quantity of information. When you combine single-cell RNA-seq with flow cytometry through artificial intelligence, the computer can tell us whether we can see significant differences between MS patients and healthy donors. And I'll show you how this works. So in CanProCo, we have 4 sub-cohorts, which are made up of 150 The first subcohort is made up of 150 patients with the cyclical form of SP. No, pardon me. The first cohort is 150 patients with isolated radiological syndromes. Those are people who have resonances that are typical of MS, but never had symptoms and have no disability. They're like pre-MS patients. Those isolated radiological syndromes. We have 200 patients having the cyclical form of the disease, relapse remissions, and that had not been treated when they were recruited. We have 100 patients with a very rare form of multiple sclerosis, which is called the primary progressive form, and we have some 50 healthy controls. And those are the patients that from which we sampled blood as right from the beginning of the disease, and we will follow them during 7 years to be able to associate the trajectory of the disease with the initial blood sample in single-cell RNA-seq and in flow cytometry. So we did a pilot experiment 1.5 years ago in which we took patients with multiple sclerosis and some healthy controls, on which we took blood, on which we did single-cell RNA-seq and flow cytometry to identify the cell subtypes present in their blood, and then to analyze them using artificial intelligence. I will spare you the names of those markers. This is in flow cytometry. We created panels of cell markers that allowed us to identify all the large populations of cells you can find in the blood, namely the T cells and all their variations, the B cells, the NK cells, which are the killer cells, and the cells of the myeloid line, which are essentially macrophages. Flow cytometry combined with artificial intelligence led us to a great surprise. The computer, after analyzing the panels of healthy controls and MS patients, was able to determine that some cell populations were overrepresented in the blood of MS patients, while other cell populations were overrepresented in the blood of the healthy controls. And this is without us having to do anything. It's really the computer that determines if there are any differences that are sufficiently significant between the patients and the healthy controls in order to determine that they are sufficiently different to be actually considered as statistically different. I'll give you an example here. You see that each point here, this is the blood of one donor, one control donor, or one healthy donor, and one donor with multiple sclerosis. You see that each point here is a cell, and the cells are grouped into families, into populations, depending on the markers that they express at their cell surface in flow cytometry. You see that there are a lot more cells like this in healthy donors than in patients with multiple sclerosis. When we ask the computer, what is this population? The computer tells us, well, those are the cells that express CD16 and CD56. Those are NK cells, killer cells. A second example, you see here this population, which is a lot less numerous in healthy controls than in patients with MS. And when we ask the computer to tell us what is this population, the computer tells us those are CD8 T cells, memory effectors. So there's a lot more memory effector T cells in the patients with MS. But will the flow cytometry data will they be confirmed by the scRNA-seq? That's what we wanted to establish. So we did single-cell RNA-seq on the same samples. Now we will not use flow cytometry. We'll look at all the RNAs expressed by both patients and healthy donors. And when we look at this, here again, each point is a cell, and each cell cells have been grouped into families depending on the types of RNAs that are expressed by those cells. The closer the RNAs are close to one another, the more there will be grouped together. That's one family of blood cells. That's another family of blood cells. This is the third family of blood cells. These cells are extremely different from those cells. And when we put charts like this, if we compare healthy controls and we superimpose this on top of patients with MS, we see that here in pink, some populations are made up essentially of people with MS. Here, there, and there. So we ask the computer again, identify for me what are those cells that are more present in MS patients, and the computer tells us, okay, those cells here are the NK cells, those are the CD8 T cells, these are the CD4 T cells, and the computer identifies all the populations of CD4 T cells and all the blood populations, both in affected patients and healthy donors. And what do we see? Well, we see that CD8 T cells here, CD8 T cells, which we saw increased in the blood of affected patients through flow cytometry, are also increased when we look at them through scRNA-seq in patients with MS who have MS. Similarly, the killer cells, the NK cells, are increased in single-cell RNA-seq in the blood of healthy controls. They have more killer cells when we compare with the blood of patients with MS. So for us, that was a crucial step, which allows us to say now that the approaches we decided upon with CanProCo with all my colleagues, which is to study the blood of the patients, was valid. So, in conclusion, combining flow cytometry and single-cell RNA-seq allows us to identify white cell populations that are different between MS patients and controls. With the 500 patients recruited into CanProCo, we will be able to identify markers that predict the good trajectory or a bad trajectory in terms of progression that could affect our patients. But that will be a very long-term work. That will take 5 or 10 years because we have to determine 10 years from now how the patients recruited in CanProCo did. What will happen to them? Will some of them progress? Because only then we will be able to get back to the baseline initial analysis and to look for differences between those who progressed and those who did not progress. To conclude, I think I have to thank the MS Society of Canada for this wonderful initiative that is CanProCo. It's the MS Society of Canada that finances, as well as a couple of pharmaceuticals. They are the ones who finance CanProCo. That was an initiative of the society 4 or 5 years ago, and we all helped the society to conduct that study. I would like to thank people who worked in my lab to generate all those data that will allow us to do the analysis of CanProCo, and all the members of our lab, as well as members of the clinic, who are working very hard for MS patients in Montreal. On this, I wish you a great end of evening, and I think there will be a question period.
This was very interesting to hear you on these complex subjects, topics. So this was pre-registered in order to ensure that there would be no technical bugs coming up. So we will now invite you to ask your questions, and you can please write your questions in the chat box. And while we wait, Dr. Prat, can you talk about the COVID-19 and how it affects your day-to-day work and the pace of your progress? Well, first of all, the first impact is that tonight I'm talking to you from in a quarantine from my cottage because my son contracted COVID, the Brazilian variant. So we're isolated in our cottage. The second impact is mainly with patient care, the mode of communication. During the past year, we changed our modes of communication. For sure, the first visit, the first diagnostics, we prefer to do them in person. So there's a certain risk there. But after with many of my patients and from the SHUM patients, we are able now to communicate through video conference or telephone. So this has changed. Unfortunately, well, sometimes it's good because it's quicker when than when the patients come in. But for one reason or not, when the patients are, it's a little bit more complicated when they're not feeling well. At the scientific level, you know that all the main steps or the main developments, scientific developments are done in emergency. We're talking about the invention of submarines at the end of the First World War, and at the end of the Second World War was the atomic bomb. So a lot of the technological developments are done under emergency, and COVID has had an impact on our careers. We developed many scientific modes in the past year, which will be extremely interesting for the study of different autoimmune diseases like MS and also the psoriasis and other diseases. We did a lot of, we received a lot of funding from the federal for our research because of COVID, and the impact of this, this allowed us to do a lot of advancement where we're able to study a lot of biomarkers, molecular cellular markers by flow cytometry. And there are many benefits during this COVID episode in scientific terms. Did this affect our study for MS? No, not really. We continue to do the same projects as we had for many years, just as before for MS. I can see that there's some questions at the bottom here. Yes, I will ask you. I will read them. Oh, I can't read. It's hidden by. Okay, I will ask them. Is there an age where we can stop the modification treatments for the disease? This is an excellent question. No, there's not really an age. If there's a benefit, as long as there's a benefit for the patient, I think the treatment should not be stopped. All the studies, technical studies of research in pharmacology were done most of the time with patients between 18 and 55 or 60 years old. So we don't know if there's a real benefit after 60 years old. But we know that when we stop the treatment, many times there is what we call a breakdown of disease activity and a resurgence of the disease. And this is something that we want to avoid. So, in my opinion, even this does not adhere, or if my opinion does not go according to the ruling of the government for refunding, even if, in my opinion, if a patient is in a wheelchair because they have lost the usage of their lower limbs, I will not stop the treatment for the disease in order to conserve the superior limbs, and also the cranial nerves, so the vision, and also glutation. And also, as much as possible, I'm trying to convince the insurance companies to continue to reimburse treatments and the government to continue to reimburse treatments in order to preserve the usage of superior limbs, the arms, and the cranial nerves. In order to specify what you're saying, are you talking about the cyclical form, which brings invalidity, or somebody who has these limits of their legs in order to preserve the upper part of the body? Are you recommending to continue a treatment even in a progressive form? When we talk about a patient with a cyclical form, or they must use the wheelchair, so we say that most of the time that they have achieved the secondary form, progressive form. And so we know that the governments and the insurance companies, they don't want to reimburse the medication because it costs a lot of money for the patients who have this form, secondary progressive. But even though the patient has achieved this form, what I do and what many of my colleagues in the MS do and neurology, and what they do is that they back them with the reimbursement so that the patients can benefit the treatment in order to protect the functions in the arms and the hands and also of the cranial nerves. I understand. Thank you. We have received another question. You mentioned this, but what are the results of your research to help people who have the secondary progressive form of the disease? Well, the pro-co, they do not, for the following reason, what we're trying to determine is what are the determinants which will lead to the secondary. So we're trying to recruit people who have the cyclical form and to observe them for 7 years and to determine if initially at the beginning we are able to find things which would be a signature of the upcoming of the secondary. So the secondary progressive form, they will have the benefit, these people, of this project, and also the identification of the different factors which has led to their secondary progressive form. And the same thing for the primary progressive form also. Thank you. But there's medication that works for the secondary progressive form. We must not forget that there are different medications that can work in order to mitigate and also to people who have the secondary form at the beginning. They can also receive a medication, it's called mesantrone, and there's certain risk like all medication, but it's very efficient in order to control the secondary form. So there's options for those who have the secondary form, progressive form. Very good, thank you. There's no other questions right now in the chat, but I can ask you a question for the secondary progressive form and the medication that you're talking about. In your presentation, I thought I saw that there were certain medication, and I hear that some of them have secondary effects. Well, according to your presentation, is the risk, is it worthwhile for the result? Well, that's a good question. It's a good question. So this depends on, it depends on each patient. And each time I talk about this with patients, I say the same thing. I tell them the same thing. There are some people who say, I don't want to end up in a wheelchair. I want to take all the possible risk in order to not end up in a wheelchair. So these people, I say to them, well, the options, there are certain risks, therapeutic risks that you have. And some people, I'm sorry, it doesn't bother them very much. I'm sorry, there was a cutoff there. So I prefer, and who prefer not to have secondary effects, and so who prefer. So we have to be able to take the decision for ourselves. It's not the doctor who, the physician, the doctor who, but the patient who is facing this risk, who has to make that decision. All right, thank you. Are you able to talk about the medication? I'm sorry for the pronunciation. Misent for the active secondary progressive form. Well, this is one of the therapeutic options for the secondary progressive form. The clinical tests are convincing, and the patients who wish to have Mizen for the secondary form have a right to ask for it. I don't think that a platform discussion here for MS is a good place to talk about specific medication. But this is a medication which is approved and can be used very well, like I said, like mesotantran. In certain cases, also other medication. I understand very well your answer, and that's what my question. Can you talk about? So I'm looking at the next one. If I want to participate in the research camp, is it still possible to do it? And if yes, how? Very good question again. There's still some centers who are doing recruitment, but of course, because of COVID, we're in a slow mode with the recruitment of patients. Some centers were more at a slow pace than others because of the technical restrictions, and some centers, the research centers were closed, and some who were received an order not to receive patients. So, but we're still recruiting. In the SHUM, probably we're close to finishing recruitment, but there's still recruitment in Toronto, I think, and also in Calgary and Edmonton, and also some recruitments in British Columbia and Vancouver. But I think there's still time to manifest if you want to be recruited in Montreal. So on the website, you will find the different websites of our associates in research. R is Eleanor Roger, and she is the one that does the screening for recruitment. And so she's at the SHUM. If you think that you could be a candidate, so to be a candidate for recruitment, and we have to receive a primary progressive form for less than 10 years, and for a diagnosis for recruitment in a cyclical form or remission, we have to have a diagnosis of less than 5 years and have never been treated. If you think that you are a candidate, you can call Eleanor Roger at the SHUM, and we will send you a questionnaire and recruit you with pleasure. We have a specific question here. Have you worked with men have MS with extreme fatigue? Do you have men in your research, your studies with extreme fatigue? Yes, we have some. There are also women. I don't know. I don't think that the fatigue, extreme fatigue associated to MS is more severe in the men than women. We have some women who have a lot extreme fatigue, and yes, some are included in our research, of course. For some time now, we were able to control a little bit better the fatigue with medication that we didn't think before that could control fatigue, like Ritalin and all the derivatives of Ritalin that addresses these issues of extreme fatigue. Thank you. Very interesting here question regarding your sphere of expertise. The question just disappeared, but I thought that the T cells. I'm sorry, there's a technical problem. The question disappeared here. Well, I saw the question. I was able to read it also. Can you repeat it? Well, we're thinking that T cells were not good for MS and that they were destroying the myelin, but our research shows that there's a reduction of T cells. There's many types of T cells. When I presented these works that I just presented, I presented them in Calgary, well, before COVID, so probably November or December 2019. the elderly amongst us, the seniors amongst us. There's a study from 1990 that shows that MS, there is a deficit of NK cells. The NK cells are cells that we call the killing cells for MS, and they're lymphocytes which kill targets that could kill effectively the androcytes or neurons. But essentially, these are cells which are involved in the termination of the inflammatory activity, because these are the last ones to come in the tissue, and they kill the lymphocytes that do harm. And so if we put all these puzzle parts together, we can think that the patients who have MS have a deficiency of the NK, the killing cells. Maybe they're not able to, and it transforms and may not be able to be stopped. And so this was suggested in the past, which was forgotten or neglected, but could come back in the scientific framework. Thank you very much. What is coming up regarding cyclical treatments? What's coming regarding cyclical treatments? It's a good question. There's many molecules who are, as we say, in the pipeline, who are being evaluated. There are new formulations of molecules which target the B lymphocytes, which are de-injectable, like the interferons or Compaxone, but injectable for 2 weeks. And this is promising. There's also what we call BTKI, these emitters of tyrosine kinase breton, which is a molecule which is involved in the maturation of B lymphocytes. So this could be very interesting. Also, there's many companies who have molecules that target this pathway. I think the therapeutic arsenal for MS is very interesting, is becoming very interesting. Of course, this brings complexity for the follow-up of patients, because certain patients, we need a follow-up. So this will bring complexity, the supervision of patients. And so patients will have to be a little bit more responsible, accountable regarding their own treatment, in the sense that patients must learn that it's absolutely essential to do their blood test every month, 2 months when we're under treatment, and to do their scan in MRI, because at the present current rhythm, the treatment of MS will need a lot of resources to do all this follow-up. Thank you very much. And this ends our question period. I want to thank you for your time and for these excellent information points. Thank you also for everybody for having participated to this session. Thank you to all the speakers for giving us exceptional, outstanding, and interesting presentations. We are very grateful to all of you. I encourage you to visit the MS Society website if you are interested in learning more about the programs and services offered by the organization, or the research that are funded. I would also like to point out that the MS Society Knowledge Network is an outstanding service led by a team of staff who provide consistent, reliable, and high quality information and support services. If you have any questions, you can contact the MS Info agents Monday to Friday from 9 a.m. to 5 p.m. by phone, email, or through our chat platform. Their services are available in English and French. I would also like to express my sincere thanks to the sponsors who made this event possible. Thank you to our our main sponsor, Bristol Mayor Squibb, and to our supporting sponsors, Biogen Canada, Novartis Canada, and Rush Canada. Their support has allowed us to hear about world-class research that will help improve the quality of life for people living with MS in this country. Before we move on with the program, I invite you to take part in the MS Awareness Month by participating in our second virtual distribution of carnations on May 5th. So join us and show your support for the MS community on social media using the hashtag MS Priority SP. I also hope that you will join me on May 30th for the MS Walk this year, which coincides with the World MS Day. From coast to coast, tens of thousands of people will be walking down the streets of their neighborhoods and joining together online to show people in our country affected by MS that they are not alone. So to learn more about this event, so visit mssociety.ca. So in a few minutes, we will have the Living Well with MS workshop. So for people affected by MS from across the country. Tomorrow's agenda includes another research conference. This one will focus on MS in newcomers or immigrants to Canada. There will also be an exercise session focusing on stretching, as well as a panel discussion with experts in nutrition, physical activity, and mental health. So it's time now to take a short break before the discussion workshop begins. Please note that you do not have to log out if you wish to participate in this workshop. The same link will be used for this activity. So I invite you to take advantage of this break to stretch your legs and to take a glass of water and do some stretching while waiting for other participants to join us. So we'll see you in about 10 minutes, and thank you very much for your presence.